Yeah, okay, something like this. Yep, okay, just draw something like this. The largest organelles, yeah, the largest cell structure, which is your nucleus. Okay, remember to make some holes. Okay, in the nucleus, you have nuclear membrane, and then there is holes. We call them as nuclear pore. Later I will label for you guys. Okay, you have this black region, a black hole in the nucleus, yeah? Okay, later I will label for you guys. And then you have DNA strand in the nucleus. Okay, so come, we label this. So in nucleus, the whole thing is the nucleus. But in A levels, you will learn that nucleus has different structure. Okay, so you have the outer layer, we call them as the nuclear envelope. Or nuclear membrane also can, yeah, either one. Nuclear envelope. Yeah, okay. So the hole, remember, yeah, it's called nuclear pore. Okay, and what is the black thingy called? This is called nucleolus. Okay, nucleolus. And the strand, yeah, is your DNA strand. But in this part, we don't call them as chromosome. We call them as chromatin. Okay, what's the difference? Chromatin is uncoiled chromosome. Okay, guys, look in front for a bit. Don't copy first, yeah? Okay, so what happened is, in chromosome, remember you learned in IG? It's like a ribbon shape, ma. It's like a ribbon shape. This is your chromosome. So what is actually a chromosome? Chromosome is where you coil the DNA. Okay, if you zoom in, you will see something like this. You coil the DNA, you get the chromosome. So if you uncoil them, if you pull them up, or you straighten them, you will get your chromatin, no? Okay? So the chromatin is the uncoiled chromosome. If you straighten the chromosome, you get the chromatin. Alright? Okay, done. So you can add the description at the bottom, yeah? I write down for you guys. Chromatin is uncoiled chromosome. Yeah? Okay, good. So the whole thing will made up your what? Made up your nucleus. Okay, so the whole thing is your nucleus. Ta-da! So how many things you need to know in your nucleus? 4 structure. Nuclear envelope, nuclear pore, nucleolus, the black region, the dark hole, the black hole, and the last one is the chromatin. Okay, now, so as usual, like what you learned in IG, we can continue drawing it, yeah? Okay, I think we draw first. So you draw everything first, and then I will label one by one. Alright, can Okay, so one more last one. So this one is curved a bit. Curved and shorter, yeah? Okay, if you see the comparison, this one is curved as compared to whatever that we draw just now. I promise you lah. Seriously, if you draw right in biology, right, some of the part you can remember easily. Okay, you don't even need to memorize because as you draw, it goes into your brain. I tell you, some of your school don't even bother to teach all this stuff, yeah. So if you can, just try to absorb as much as you can today. Okay, your teacher will just say, okay, you guys can study this on your own lah. Can? Okay, come. Let's look in front first. Okay, guys, can you guys tell me, we label together, yeah? What is this structure called? Remember, if it's attached to the nucleus, RER, correct. Okay, so RER, your rough endoplasmic reticulum. Okay, so the one that attached to it, it will be your ribosome. Okay, I want to emphasize this, guys. Can you see? There is 2 ribosomes. One is attached to the RER, the other one is in the cytoplasm. Okay, so this is your ribosome. Can you guys add on for me? Ribosome, you got 2 options. Okay, there is 70S and 80S. It's just the size of it. So for eukaryotes, it's called 80S ribosome. Is it important to remember? Very important. Because they always ask you guys to compare with the bacteria, the prokaryote. The prokaryote has 70S. Eukaryote, which is the animal and plant cells, has 80S. Remember that. Okay? So next one, it will be your SER. Okay, your smooth endoplasmic reticulum. Smooth endoplasmic reticulum. Okay, your SER. So you see, the shape of SER and RER is almost the same. The differences is RER has ribosome. Okay? This one, guys, tell me, what is this structure? What is meant by ATS? It's just the size of it. Okay, the size. We call them the subunit. Yes, mitochondria, yeah. This is your mitochondria. Okay, very good. Correct. Okay, so this one is new for you guys. It's known as lysosome. I will explain the importance of lysosome. Okay, inside lysosome, you will get enzyme. So do you see the dot inside? Is known as your digestive enzyme. Okay, so your lysosome is actually the cleaner in the cells. Help you to digest the bacteria. Okay, or unwanted material will be digested by the lysosome. Alright, okay guys, come. What is this? New for you guys as well. This one and this one, okay, is very easy to recognize, guys. Listen, yeah. Golgi apparatus is always curved. Okay, whenever you see they are curved, it's definitely Golgi apparatus. Clear? Because in higher levels, they will ask you to see the microscopic view. Okay, the microscopic view allow you to identify them. Remember, it is curved. Okay, so this is your Golgi apparatus. Okay, done. Alright guys, so what is the circle thingy? That is your vesicle. So we have two types of vesicle, yeah? Okay, write it down. So this is your transport vesicle. And the one that going out is your secretory vesicle. Okay, let me explain. Look here, guys. Secretory means what? Secret ma. So this secretory vesicle will move to the cell membrane. Okay, and later on, it will fuse with the cell membrane to secrete the content. Okay, I draw again for you guys, yeah? Look here. So this fella, right, it will move to the membrane. Okay, so after that, it will fuse with the membrane and releases the content. Hence the name, secretory vesicle, because it is to secrete the content out of the cells. So what is inside the vesicle? Usually it's your protein, your enzymes, your hormones, your antibodies. Okay, all right. Can So remember, yeah, you have two types of vesicle. Transport is to carry. So this is to carry from here to here. Can you guys see that? Secrete means that, secretory means that it will secrete the content out of the cells. Hence the difference in the name. Clear? Okay, all right. Okay, last one. Last one is also new for you guys. These fella, they have two of them, yeah? They are in how to differentiate vesicle and lysosome. Good question, very good. Usually, lysosome are bigger. Okay, usually lysosome are bigger. Understand? But in exam, right, they won't be so bad one. They will just tell you it's lysosome. Usually, they won't ask you to label the lysosome. Okay, if they did, they will tell you, oh, this structure contains enzyme. So you know, it is lysosome. Understand? They will just tell you a hint. Then for you to differentiate the lysosome and the vesicle, then you'll be able to label them already. Okay, so this structure on this side, they are right angle, yeah? Okay, so we call this as centriole. Okay, this is very important. This centriole is used in cell division, which you will learn soon. Okay, it is used in cell division, yeah? Okay, done. All right. Okay, guys. One more last one. Inside your cell, right, you have a lot of, okay, we call this as the microtubule. Can you guys see that? It's like a strand. Okay. So this microtubule, right, it is to create movement. Okay. So you know, like a puppet, it's like a puppet, right? So the microtubule will help to pull. So the pulling will allow the organelles to move from one area to another area. So you guys must be thinking, hey, sure, how does the vesicle move? Due to the microtubule. Okay, so this microtubule will be like a puppet, you know, it will pull the tube. So as they pull the tube, it will move the structure inside the cells. All right, okay, I write nicely for you guys. No space. Microtubule. Okay, that's all. That's all you need to know. Okay, if you ask me, what is this? This one, you should be able to know this. This is your microvilli. Okay, this is the microvilli. Okay, any questions? So what are the new structure that you learned today? You might want to highlight. So the new one that you learned today is microtubule, right? Centriole. Okay, SER, I think some of you heard before already in IG, right? Okay, the rest also you heard before. Lysosome, it could be new for you guys. Golgi apparatus, that's all. And also the whole structure of nucleus. Okay, so the rest you learned before already. So you only need to add on 4 new things in your brain. Okay, come, let me tell you something important. Centriole can only be found in animal cells. Remember that, okay? In plant cells, do you have centriole? Don't have. Okay, clear, yeah? They always ask you also in your question. Which one only found in animal cells? Centriole. You want the screenshot? Okay, darling, no problem.